Good evening. Welcome to the live session of week 11 for the NPTEL course on environmental biotechnology. I am Guru Priya, a Prime Minister's Research Fellow in the Department of Civil Engineering, IIT Madras. I hope the screen is visible and also I am audible clearly. If there is any issue, please let me know in the chat box. So before we start today's session, I would like to ask if there are any specific questions that you would want me to address from any of the previous sessions or based on the course content. If there are any doubts, please post in the chat box so that I'll try to clear it up. Okay, so we will start with today's session. So until now, we have seen about uh, uh, all the biodegradation processes, the environmental biotechnological approach that we need to study about all these microorganisms, their roles, and how to extrapolate these roles into biological processes that help us in pollution abatement and waste removal. So, all of this we have been discussing so far. So, in today's session, again, we will start talking about this microorganisms and their role in transformation or remediation of metals. Is the screen visible and is my audio clear? Okay. So, excuse- okay. So till now we have seen about the remediation of organic pollutants. So we all know organic pollutants have the potential to undergo complete mineralization, which means the organic matters are going to be made up of mostly carbon, hydrogen, oxygen, nitrogen, sulfur, etc. And they will undergo complete mineralization, that is the conversion into carbon dioxide and hydrogen. So once it turns into, sorry, carbon dioxide and water. So once it turns out to be carbon dioxide and water, uh, it is again going to be fixed by the plants which take in this and then synthesize some organic matter which will undergo series of process, and finally when it is a waste it will again be degraded. So this will be a cyclic process, and since you are converting the organic product into something very basic, again you call this process a complete mineralization process. And this is also an oxidative mechanism where you try to oxidize the organic pollutant. But in the case of metals, they are mostly inorganic. So you cannot break down metals. So metals undergo biotransformation and not biodegradation. So here what happens is either there is some alteration of the structure of that metal based pollutant. Either the molecular or atomic structure of the compound is going to get altered, and this will further change the chemical properties. Chemical properties like the solubility, the oxidation state, and its interaction with the aqueous system or any system that is present. All these properties will be altered, and in turn the environmental behavior again will get altered. So this may render a metal more toxic or less toxic. It depends on the kind of transformation that is happening. So when we are trying to like do remediation, we are obviously trying to make the metal less toxic, and also most cases less bioavailable, and we are trying to immobilize the metal, but we will not be able to break down the metal. So the different processes that can happen could be detoxification, bioleaching, biotransformation, biosorption, bioprecipitation, or bioaccumulation. So in brief, detoxification may be like you will try to reduce the toxicity of the metal that is present, like using extracellular chelation or binding the metal to the cell wall components, or using transport channels, or during intracellular chelation. Can either be extracellular chelation or intracellular chelation. You are trying to render the metal into a less or non-toxic form. In case of bioaccumulation, the microorganism tries to take up the metal into its cell and accumulate it inside the microbial cell. This can happen via precipitation, adsorption, or and metal efflux through chelation. And in case of bioprecipitation, it is all again an immobilization technique where changing the solubilization properties or the surface properties of the metal will lead to precipitation of that metal, and hence it cannot travel through the aqueous system and reach other places. It is in turn immobilized in a particular place. In case of biosorption, it can happen on the surface of the microorganism itself, where there can be proper binding sites and binding affinity between the microbe and the metal itself, leading to the metal getting adsorbed or attached to the surface of the microorganism. And then you have this biotransformation, where a series of reactions like oxidation, reduction, hydrolysis, condensation, or isomerization can happen and change the chemical state of that metal or metal based compound. So in detoxification, we have highly specific biochemical pathways, and this naturally occur in the microorganism as protective mechanisms to deal with toxic heavy metals. We know that if there is any environment, and when the environment is very dynamic, the organism also tries to get adapted to the situation itself, right? So it tries to adapt itself. So in that process, whenever there is some toxic metal that is present in an environment, the microbe again tries to get accustomed to that environment. So during this process of evolution, it is able to develop mechanisms, protective mechanisms, in order to survive in the place where the same toxic metal is present. So this is the first point in the detoxification based method. So specifically, we have seen such protective mechanisms in microorganisms against chromium, arsenic, mercury, cadmium, cobalt, and uh, copper metals. So microbes have their biochemical systems or biochemical capability to withstand all this chromium, arsenic, mercury, cadmium, cobalt, and copper. So some specific examples include the enzymatic reduction of the specific metals. So we render it less toxic, or we can also talk about the efflux pump systems that is present in the microorganisms, or the transporters that can back and forth transport the metals within and outside the system, and then within the system or outside the system you can see some other biotransformation processes also occurring hand in hand, or you can also see bioaccumulation happening in certain microorganisms. So generally detoxification methods are naturally present in the microorganism, and if we are in need, we can also modify the microorganism or optimize the process such that we uh, enhance this detoxification process for bioremediation applications. So first we saw detoxification based method, and now we have entrapment based method, which we partially were telling like immobilization method kind of a thing in the first slide that we saw. So microbes produce new mineral phases that result in toxic metal entrapment within the soil or sediment. So if you have studied about some coagulation, flocculation process and all, something will be suspended in that in aqueous system, and when you add some coagulant, you will change the solubility properties of the suspended particle. It will settle. So this is the mechanism of flocculation. When you talk about the entrapment method here, there are some substances that is being produced by the microorganism which is released into the aqueous system. It kind of entraps the metal in that mineral phase itself. Therefore, with the aqueous system, the metal is not going to get carried away or it's not going to move. It's going to get settled in that soil or sediment. So this method generally target broad categories of metals like divalent cations, and the process leads to metal immobilization in environmental components. Particularly, it is effective for positively charged metal species. So it, especially divalent cations, this entrapment method is useful. And the last one is the mobilization or immobilization based method. So here the production of there is production of the chemical moieties which can form complex with the toxic metal. So this includes phosphates, carbonates, hydroxyl ions, sulfide ligands from the microbe. So they will again form a ligand donor interaction between the this group and that metal, and it can result in either metal mobilization or immobilization. So in some cases you will need immobilization just to contain the metal in that location itself. In some cases you will need to mobilize whenever there is the metal is trapped somewhere. You need to have measures to mobilize so that you recover it from that system itself. So you can either have mobilization or immobilization. The soluble metal complexes can be pumped out and recovered, while insoluble precipitates provide the containment. So next, talking about the bioaccumulation, it involves the active transport of these metal ions inside the cell, and for this they use specific or general transporters. So there are many general transporters present in the microorganism. General transporters mean we all know that metals like calcium, sodium, magnesium are all essential trace elements or micronutrients. So there are different categories in which the organism will need all these ions, elements or metals, right? So in a similar way, there are transporters, regulated transporters that carry like sodium from within the cell to outside, or potassium from outside the cell to within. So there are specific transporters in microorganisms. So these transporters can be general or specific, and that can be present in the microorganism, which will also facilitate the active transport of the metals also. So active transport, in the sense there is energy requirement in active transport. So there are two types of transport. One is the active, another one is the passive transport. So passive transport is generally, let's say, it will follow a concentration gradient from a more concentrated region to a less concentrated region. When there is a travel, you need not like spend any energy in that travel. It naturally flows from the highly concentrated site to the less concentrated site. Whereas in case of active transport, you will need the expenditure of some energy. Let's say we are traveling against a concentration gradient, then the transport is not going to be natural. You will spend some energy to bring the ions from one end to the other end. So therefore this is energy dependent, where the adenosine triphosphate molecules, which is the power currency of the cell, is going to be utilized. And it can utilize specific metal transporters. For example, zinc, nickel and copper have specific transporters which can transport these metals, and it requires active cells for sufficient energy supply. So if the microorganism is able to produce a large amount of energy, then this process is going to be favored. If the microorganism itself is like not having enough energy for its survival, then I don't think it can do this transport. So one basic condition is the this transport requires energy, and hence active cells can only go about doing this bioaccumulation. And another important point is that we cannot keep on accumulating infinite number of metal ions or metals into the microorganism. There is certain limit. So we can only accumulate to a certain concentration level. After that, the microorganism starts employing detoxification strategies to get rid of the negative effects from these metals. So in case of bioaccumulation, yeah, it can handle divalent cations, arsenate and chromate ions. For example, we have pseudomonas species that can accumulate uranium through polyphosphate granules. So the energy expenditure and the amount of accumulation are the two important things that you have to remember when you talk about bioaccumulation. So what if the accumulation goes beyond a tolerance limit? Then the cell starts implementing the detoxification strategy. So the detoxification strategies include efflux systems, that is pumping out the metal out of the system. So first what it is doing, it is accumulating. Again it is pumping out. So that is efflux system, and it is also an active export and requires concentration gradient or energy expenditure. Next, you can see that there is compartmentalization, where the metal is sequestered in specific regions, for example in the cytoplasm, for example in the periplasm. So the cell decides one particular location, the metal is being sequestered only in that location. And the third one is the enzymatic conversion, where the metal is enzymatically converted into some other form. It is reduced, like for example, the oxidation state is changed and the metal is reduced, and generally reduced metals are less toxic than their oxidated counterparts. And finally, you have one more mechanism, which is the metallothionein production. So the metallothionein is a small protein molecule, and it helps in the metal sequestration. We'll see about this in the upcoming slides as well. So we talked about bioaccumulation. Now we'll talk about biosorption. So in case of biosorption, it's a metabolism independent physical physicochemical process involving the adsorption of that metal ion onto the cell surface. So as said, for bioaccumulation, it is metabolism dependent because it requires energy in the form of ATP, and only active cells can perform bioaccumulation. So metabolically active cells will have enough energy to perform bioaccumulation. Whereas in the case of biosorption, it is metabolism independent. It is just a physicochemical process where the metal ion is going to get attached to the microbial surface. So here you need not have any expenditure of ATP or energy. Therefore, it's a metabolism independent process, and it is mediated by the negatively charged cell surface groups like carboxyl groups, amine groups, hydroxyls, phosphates, and sulfhydryl groups. So in the cell surface of the microorganism, you can find all these negatively charged groups. So it is very effective in remediating sites with uh, positively charged metal contamination. And here, since it is metabolism independent, you just have used the microorganism as a medium to attach some metals, right? So in that case, it can happen in both live and dead cells. Because there is energy expenditure required, bioaccumulation can happen only with actively growing cells, whereas biosorption can take place because of live cells as well as dead cells. And this process has a rapid kinetics and high surface area to volume ratio, because there is comparing the size of the metal ion and the surface of the cell membrane of the microorganism. It is a high surface area to volume ratio process, and it has a rapid kinetics. So here it can involve electrostatic adsorption. Electrostatic in the sense positively charged ions getting attracted to the negatively charged cell wall or membrane, and then there happens a microprecipitate. There can also be the presence of microprecipitation and ion exchange happening in the process. So the advantages of this BIOS process includes that it is more effective than physicochemical methods at low concentration. So in low concentration, whenever you use a normal adsorbent which is of a non-biological nature, then you won't get that much efficiency, removal efficiency, whereas biosorption is more effective, and it can use low cost waste biomasses. It has rapid kinetics, and the biomass is regenerable, so you can regenerate the biomass. It has higher specificity than ion exchange resins, and you can also work on genetically engineering the microorganisms so that to enhance their adsorption specificity and efficiency or the entire performance. So you can do genetic engineering targeting these benefits also. But the disadvantages on the other hand include that desorption is also as rapid as this option. So we saw that the biosorption has rapid kinetics, but equally possible that the desorption is also so rapid so that the net gain is nothing for the person who is doing the bioremediation. And also we talked about the usage of both live and dead cells. In case of dead cells, sometimes we can expect the metal re-release into the system and decomposition of the microbial cell, because that is a organic matter. And there will be a high competition from other cations for binding sites, not only the target ions that we are trying to remediate the system from. Also there will be competition from other cations, and there will be quick saturation of the cell surfaces. So you have to recover it from the system, desorb it, and again do adsorption. This has to be really very rapid and quick, because the saturation can be very quick. And the major disadvantage is that this may not be able to produce adequate long-term bioremediation in uh, in situ contaminated sites. So these are the advantages and disadvantages of the biosorption process. So just to have a quick comparison of bioaccumulation and biosorption, we could see that there is energy requirement for bioaccumulation to occur, whereas in case of biosorption, it is very low. And in case of bioaccumulation, we can achieve bioaccumulation only using live cells, whereas biosorption can happen due to live and dead cells. And the mechanism is going to be active transport in case of bioaccumulation, whereas it is a physicochemical adsorption in the case of biosorption. And bioaccumulation is highly specific compared to biosorption, and the capacity for bioaccumulation is limited by the concentration of the metal, which renders it toxic to the microorganism, whereas in biosorption the adsorption capacity can be usually higher. And reversibility, it is controlled by cellular processes. Biosorption can be rapidly reversible. So next comes the bioreduction. So bioreduction involves the enzymatic or dissimilatory reduction of metals from higher to lower oxidation states, and this results in insoluble precipitate formation and precipitation. So for example, we have the reduction of chromium 6 to chromium 3, arsenic 5 to arsenic 3. So what happens is we are just lowering the oxidation state, and hence the metal forms precipitates. So in the case of this reduction can happen via direct enzymatic reduction. So in direct enzymatic reduction, the microorganisms will use uh, cytochrome based or flavin based systems to transfer electrons from organic electron donor to metal species. So here what happens, you will have a organic matter which will act as a electron and carbon source for the microorganism. So by metabolizing that, the microorganism will generate electron and carbon, and using cytochrome and flavin based systems, this electron that is sequestered from the organic substrate will be used to reduce the metal. So the key factor here is the availability of the organic carbon source and electron donor source. And this is highly specific enzymes for metals are not required by cells. It is used in engineered dinococons radiodurans for radioactive environments. And in case of mercury reduction by mercuric reductase and arsenic reduction by arsenate reductase, all these are examples of direct enzymatic reduction. In case of indirect enzymatic reduction, it involves the uh, it involves the use of intermediate electron carriers that will facilitate the metal reduction. For example, ion mediated reduction, where Fe3 plus is converted to Fe2 plus, it is reduced by ion reductases, followed by the Fe2 plus reducing bacteria. I think there is a typo here. I'm sorry for that. So first you will have the reduction of Fe3 plus to Fe2 plus, and further Fe2 plus will be reduced. This can also be seen in sulfide mediated reduction, where sulfate reducing bacteria produces hydrogen sulfide, which then reduces the metal species. And the general mechanism will include using periplasmic hydrogenases and cytochrome systems. The advantage is that it can handle multiple metals using a single electron carrying system itself. So this is about the indirect enzymatic reduction. And next you have another type of bioreduction, which is called the dissimilatory metal reduction. So unlike the specific enzymatic reduction, dissimilatory process uses metals as the terminal electron acceptors during anaerobic metabolism. So here there is going to be an electron donor, and this metal will be the final electron, terminal electron acceptor. And this generally happens in anaerobic environments. And these reduced products are not utilized by the microorganisms, and hence you call them as dissimilatory process. For example, chromium, uranium, technetium reduction happening under anaerobic reduction is via the dissimilatory metal reduction pathway. And even for this, you will need organic carbon source and appropriate redox conditions for this process to be viable using microorganisms. And when we talk about biomineralization in the context of metals, it involves the precipitation of metals through microbially generated ligands, creating stable mineral deposits. For example, you have this phosphate mediated biomineralization where bacteria produces phosphatase enzymes. This is going to release the phosphate from organic phosphate compounds, and the phosphate ligand hence generated binds with the divalent metal ions and forms a insoluble metal phosphate complex. So you can find this in the Citrobacter species, which produces uranium phosphate and europium deposits, and this is can be generally used in uh, remediation of radionuclides and heavy metals. There is one more mechanism, which is the sulfide mediated biomineralization, where sulfate reducing bacteria convert sulfate SO4 minus to hydrogen sulfide. And this hydrogen sulfide reacts with the divalent cations to form metal sulfide. So this forms mineral nucleation centers, and finally they grow into large deposits. So the examples of sulfide mediated biomineralization include the formation of cadmium sulfide, zinc sulfide, and copper sulfide minerals. And next, when we talk about the precipitation and immobilization, we will see how this is happening. First is the nucleation and growth phase. So, initially, when the precipitation is happening, that is called the nucleation center, and as it expands, there is going to be gradual mineral growth and will create a large deposit, and this will result in the long term metal immobilization. So the environmental stability aspect in this precipitation and immobilization is that precipitates will remain stable under neutral pH conditions, but when you alter the pH, there is chances of again it being mobilized, and the reduced forms will have low solubility and bioavailability because it is reduced and it is formed a deposit. You can assume that it's going to have low solubility, and henceforth the bioavailability is also going to be very less. It So while doing it on the field, it will create a protective coating and low permeability barrier. So up to this, we have seen about the mechanisms. Please let me know if there is any doubt in this part so far. If so, you can ask in the chat box. I'll just wait for a minute. Okay. So now we'll see about the role of metallothioneins in microbial metal tolerance. So initially we saw few factors, one of which was metallothioneins. Metallothioneins represent a crucial family of metal chelating proteins. So they are basically proteins that has the ability to chelate the metal ions, and this helps in uh, in inducing metal tolerance in microorganisms, and they have become valuable tools in genetic engineering for enhanced bioremediation applications. So generally, as we saw, metallothioneins are proteins which have low molecular weight, and cysteine is an amino acid. So proteins are generally made of amino acid, and this metallothioneins is a cysteine-rich protein. So it contains high concentrations of sulfhydryl groups, uh, which is representative when we talk about cysteine. Cysteine is a sulfide containing amino acid, right? So because this protein is cysteine rich, it's going to have high concentrations of sulfhydryl groups, and it can form metal sulfur bonds with divalent metal cations. It's primarily found in the eukaryotic organisms and cyanobacteria, and its primary function is to regulate the intracellular metal concentrations and provide detoxification. So now we have an idea of what is metallothioneins. Metallothioneins are low molecular weight cysteine rich proteins. So cysteine rich again implies that they are rich in sulfhydryl groups which can bond with divalent metal cations, and the biological role of them is to regulate the intracellular metal concentrations and provide detoxification. Okay. So now we will see about the enzymatic transformations of metals and metalloids. So enzymatic transformation again is a sophisticated microbial strategy because the microorganism should be fully capable, evolved enough such that it synthesizes enzymes and helps in the transformation of metals and metalloids. Even though the metal is not very essential for a cellular metabolism of the microbe itself. And these processes are not connected to the electron transport or disseminatory reduction for energy generation. Instead, they involve specific enzymes that have evolved solely for detoxification. So here we are not going to talk about any electron transport or disseminatory reduction, and there is, because we are not going to talk about that, there is no space for talking about energy generation also. This is just the microorganism trying to only detoxify but get nothing out of it uh, in return. So you use specific enzymes evolved solely for detoxification, and this aids in the conversion of the toxic forms to less toxic or removable form of that species from the system itself. And this is to confer protection of cellular macromolecules from the metal interference and to maintain the homeostasis in metal contaminated environment. For example, you have this mercury or MER operand system where the complete gene cluster encoding entire mercury resistant system is present, and it has a coordinated expression of transport and detoxification components. And there is also a special or specific transporter for facilitating the uptake of mercury alone. So it has two components. One is the mercury transporter and another is a mercury reductase enzyme. So in the case of mercury transport, there is a specific transporter and it is highly selective due to extreme toxicity of mercury, and it facilitates rapid transport to minimize the cellular exposure time. So if the cell is going to be exposed more, then there's going to be toxic effect on the cell itself. And on the other hand, you have this mercuric reductase, which is coded by MER-A gene, and it is a cytosolic enzyme, which is a cytoplasmic enzyme, and it is responsible for mercury reduction. Uh, so it is going to reduce Hg2 plus to uh, Hg0, which is the elemental mercury. So the whole idea here is that the mercury with the zero oxidation state is volatile and will diffuse out of the system. So you have successfully eliminated it out of the system. Again, this process is going to be NADPH dependent reduction process. So there is going to be some amount of energy expenditure. Next, we'll talk about the biogenic minerals. So biogenic minerals are microbially produced mineral phases. They are produced by the microbial metabolic processes, and therefore they are very distinct from the minerals that are geochemically formed. So the whole idea is that these materials are going to combine the selectivity and specificity of biological systems with the stability and reactivity of mineral phases. So can you tell me what is the difference between selectivity and specificity? Okay, the fundamental difference between selectivity and specificity, they are uh, what it is similarly sounding terms, but they are not the same. When you have two to three things, when you prefer one over the other, it is selectivity. But when you are going to only choose one specifically, that is known as specificity. Like if you prefer A more than B is selectivity, you will prefer only A is specificity. So biological systems have this selectivity and specificity, and this mineral phases are going to be very stable and reactive. So these biogenic minerals are going to combine all these properties and help us in remediating the metals. So the key characteristics of biogenic minerals include that they are of biological origin. Yeah, obviously they are produced by microorganisms through metabolism, and they have enhanced reactivity because they have high surface area and also reactive sites compared to the synthetically produced minerals. And because they are biogenic, they have high environmental compatibility, and also they are biodegradable. And then the production of these biogenic minerals is sustainable, so it can be generated using local substrates and renewable processes. For example, you have manganese oxide biogenic minerals. So let's see about the biooxidation of manganese, where the substrate is going to have Mn2 plus ions. Mn2 plus in its 2 plus oxidation state is going to be solubilized, and in the process it is going to be oxidized from Mn2 plus to Mn4 plus ions. So in this process, there is going to be formation of manganese dioxide, and in this form, it is going in the oxidized form, it is going to be insoluble. So the mechanism, if you are going have to describe here, it is going to be dissimilatory biooxidation, where manganese serves as the electron acceptor. In microbial systems, you have manganese oxidizing bacteria, and these are specialized organisms capable of oxidizing Mn2 plus to Mn4 plus. And you also have iron oxidizing bacteria, and some of the iron oxidizing bacteria have the capacity to oxidize both iron as well as manganese. And the environmental condition required to produce this manganese oxide, biogenic manganese oxide, is uh, that you need aerobic to micro aerophilic condition, and the pH requirement is near neutral to slightly alkaline condition. So we have seen about how it is produced. Now we'll see about the mineral formation process. Initially, Mn2 plus is converted to Mn3 plus or Mn4 plus through the bacterial enzymes, and that is going to be the core point around which there is going to be nucleation happening, where there is going to be initial formation of MnO2 nucleation sites, and again the crystal will then grow, where the progressive mineral development around bacterial cells will happen, and finally it will stabilize, forming the crystalline manganese oxide phases. So generally, if you see the biogenic manganese oxide will have high surface area, hence it will have a high adsorption capacity, and it will also have nanoscale structure, which means we can expect high reactivity and interaction sites. So it has high surface area, high reactivity and interaction sites, and the morphology is going to be porous, and hence it can facilitate improved mass transfer and accessibility, and it's going to be chemically stable, which is it's going to be resistant to dissolution under normal environmental conditions. This is about the physical properties. In case of chemical properties, it's going to have strong oxidizing potential, and it is effective for oxidation of organic and inorganic contaminants again. And then it will have high surface reactivity, which means you can have multiple functional groups that can facilitate even more pollutant binding. It will be able to have pH buffering capacity, and we know that the surface reactivity is going to be high, it is also going to be uh, selective, which means it will have different affinity for different pollutant classes. So after this, next comes the concept of metal homeostasis. First of all, homeostasis is going to be the concept of maintenance of normal conditions in any living system. Okay, in case of metal homeostasis, it enables the cells to maintain the optimal metal concentration and protects the cell against the toxicity. So generally, it's not that metal is completely irrelevant to microorganisms or metal isn't needed at all. That's not the case. Metals in microbes can act as enzyme cofactors, structural components, electron carriers, as well as regulatory elements. So I think in the last lecture we saw that metal, in terms of the microbial physiology, the metal can be essential and non-toxic, essential and but toxic, and always toxic. We also saw some examples. Some metals are essential and non-toxic, so even if they are present at higher concentration, it's not going to affect the microbe. Some are essential and toxic, so therefore there is an optimal level below which it is, in that range it is essential. Above the optimal level, it's going to cause some toxicity to the organism. And there are some metals like mercury, which has no biological role to play in the microorganism itself. It's just going to be toxic always. So in case of metal homeostasis, the first concept that we'll be seeing is about the transporter classification. So you have both general transporters and specific transporters. In case of general transporters, the specificity is going to be broad. It will be able to transport multiple divalent cations. And when we talk about it's going to be broad, not everything can go inside. There is going to be something that's going to act as a cutoff factor, and the cutoff factor here is going to be the ionic radius dependence. So it's not about the metal or the oxidation state. What is the radius, ionic radius, that this oxidation state confers? That is going to be the cutoff factor in case of general transporters. So the selection of or the decision whether to transport the metal or not is going to be based on its size and charge similarities. For example, there are general divalent cation transporters. So the advantages of having general transporters is that it is metabolically efficient for multiple metals. So for example, there is a contaminated site and you have divalent ions. The metabolic load on the microorganism to generate different types of transporters for each of the metal is going to reduce. But the disadvantage is that it shows reduced selectivity, and it will also form a way for transporting the toxic metals into the microbial cell. In case of specific transporters, it's going to have high selectivity, which means one transporter will only transport a particular type of metal, like zinc, nickel, and copper have specific transporter. Zinc transporter only transports zinc but not copper, and vice versa, right? And it will have precise regulation. uh, it is responsive to specific metal availability and cellular needs, and there's going to be energy requirements of an ATP dependent active transport. So the examples include zinc transporter, zinc ABC transporter, nickel, and copper transporters. This is just the name of the transporter itself. And next, the cellular response to metal concentration is going to change. So when we talk about low concentration response, so we had three physiological classifications: essential and non-toxic, essential and toxic, and always toxic, right? So when we talk about this low concentration response, uh, when a metal is going to be required by the microorganism, and if it is not present, like we have vitamin deficiency, it's going to be deficient. The microorganism is going to be deficient of that metal. So in that case, you will see transporter upregulation. So you will have more copies of the transporters so that you can facilitate more transport of that metal into the system. and other alternative pathways for acquisition of that metal is can also happen, and the microorganism will start adjusting its metabolism to survive against this deficiency, and there will be some stress response like activation of metal scavenging systems. So when there is going to be optimal concentration of that metal, homeostasis will be maintained. There will be balanced regulation, which means there will be coordinated uptake of the metal and the efflux of the metal, and there will be proper storage systems uh, to store the sequestered metal. There will be a good metabolic integration where the enzyme functions and other cellular processes are going to be in harmony, and you will have regulatory feedback, which is the fine tuning of transport systems. And when you are going to have excessive concentration, which we call it as toxicity. So here the first thing that happens is the efflux is going to be activated, which efflux is sending sending the metal out of the system. So the metal is going to be exported out of the cell, and other detoxification strategies like oxidation reduction, all these processes starts functioning. Uh, these processes can happen with the aid of the enzymes, and hence it's going to be enzymatic transformation. And here the stress response is going to be activation of heat shock proteins and oxidative stress defenses. And there will be compartmentalization here, where you can see the sequestration of the metal in specific cellular region, which will aid in the efficiency of the detoxification process. So now we will see what are the advanced homeostatic strategies. One is the compartmentalization and other is the efflux system. So in case of compartmentalization, you have this the compartmentalization can happen either in the periplasm or the cytoplasm. In case of periplasm, you can see copper binding proteins in Pseudomonas. The strategy here is that the microorganism is trying to confine the metal only to the periplasmic space, which means it is protecting its cytoplasmic components, but it is still trying to figure out the optimal metal transport mechanism. The application is generally in intermediate toxicity management. It's not going to be the complete solution, but it will help you in managing the intermediate metal toxicity. In case of cytoplasmic sequestration, you will have the involvement of metallothionein systems, where these proteins will try to bind the metals, and you will have production of polyphosphate granules, and this phosphate based metal storage will happen. Again, you have other protein based systems which can act as ligand and bind to the metals. So special metal binding proteins can be generated. And finally, you have vesicles which can entrap the metal and store it in the cytoplasm. In case of efflux systems, it's going to be like we talked about in one of the previous slides, it's going to be an active transport system, which means there is energy and ATP expenditure happening. So the ATP driven pumps will be present. You will have proton antiporters, which means they will perform this metal efflux using the proton motive force based on the H plus ion concentration gradient. Your metal efflux will happen on the opposite direction, and this efflux systems can vary in a wide range between specific to broad spectrum, and the regulation of the efflux system is responsive to the intracellular metal concentration. So the stimuli or the trigger factor for efflux system to get active is the um, concentration of the metal inside the microbial cell. So these two are the advanced homeostatic strategies in microorganisms. One is compartmentalization and next one is the efflux systems. So next is about biohydrometallurgy. So in case of biohydrometallurgy, We use microorganisms to extract metals from their low grade ores. So metallurgy is basically extraction of metals from their ores. In biohydrometallurgy, we are going to use microorganisms to perform this metal extractions from low grade ores, tailings, and even from end of use waste in aqueous solution. So this is a microbially catalyzed oxidation process, and it oxidizes iron and sulfur compounds, and it results in the formation of soluble form of this metal. So ideally this process can occur in aqueous environments which can become acidic. So the microorganism should have the capability to generate acids, uh, and then the metals are solubilized into this acidic solution. So talking about the application of biohydrometallurgy, up to 15% of the global copper production and 5% of coal production is currently dependent on biohydrometallurgy. The one of the great advantages is that we take metals, convert it into some products, and throw away the products, which ends up as waste. So instead of going on for more ores, we can try to use these wastes as the systems for recovering the metals and then reuse them. So apart from copper and gold, biohydrometallurgy is also used for uranium recovery from the uranium ores. So this is just a brief history about uh, biohydrometallurgy. So starting from the 1920s to 30s, there was initial research on iron sulfur oxidizing bacteria. So this was the foundation for the studies on bioleaching. Here people only had the basic understanding of how microbial oxidation occurs, especially how microbes were able to oxidize iron and sulfur compounds. So coming to the later part, to the 1950s, the scientists were able to identify the bacterial strains prominently. So one of which is a thiobacillus species, and it was capable of uh, oxidation, oxidizing iron sulfide and copper sulfide. It was isolated and characterized. So this was one of the significant milestones in the evolution of biohydrometallurgy. In 1960s, there was development of in situ leaching of uranium and copper, along with the discovery of iron and sulfur oxidizing archaea and many other iron sulfur oxidizing bacteria. In 1980s and beyond, the setup of commercial heap leaching plants and gold extraction and gold oxidation plants for the recovery of copper, uranium, and gold in countries like USA, Chile, South Africa, and Australia was observed. So this was about commercializing. So before this, we just saw about what was the scientific or research-based evolution. Here, people started building uh, or commercializing this bioleaching. In 1990s, uh, bioleaching of copper ores and the development of bioheaps for extraction of gold, nickel, and zinc was uh, done. And later, after 2000s, the development of second and third generation biooxidation plants happened. So in current era, there is still research going on, but we have come a very far way in using microorganisms to leach metals from their ores. And this was again used as one of the strategies to combat metal contamination or metal pollutions from the wastes as well. So in bioleaching, first there is metal solubilization that should happen. So for that, there should be organic acid production. So the microorganism which is capable of performing the bioleaching should produce the organic acid, and it will interact with the insoluble metal and forms a soluble metal complexes. So this metal complex is then leached out from the ore or the waste, and it can be recovered and processed. And next you have this direct and indirect metal oxidation. So in this, there is going to be oxidation of metals, including metal sulfides and iron in particular. Metals that are in reduced and insoluble forms are oxidized, and this oxidation renders the metal s- soluble. So when it's going to be soluble, we in turn can call it as mobile, and hence they leach out from the system. So when you talk about the sulfur and sulfide oxidation, microorganisms oxidize both metal sulfides and sulfur compounds to produce sulfates. This process generates sulfuric acid and proton, and also Fe3 plus ions. So the sulfuric acid helps in leaching the metals present in the waste by solubilizing them, and the iron will act as an electron accepting molecule, further facilitating the leaching process. So as we saw about direct and indirect metal oxidation here in the second point. We'll just see a few more details about it. So in direct oxidation, the bacterial cell is in contact with the mineral surface, and it, it will directly oxidize the metal sulfide, producing the lixiviant molecules like Fe2S4, S4O3, and sulfuric acid. It can also directly oxidize metal sulfides like copper sulfide or zinc sulfide and produce soluble metal sulfates. In case of indirect, what happens is the bacterial cell generates a lixiviant without direct contact, and they use the soluble Fe2 plus ions as electron donor, oxidizing it to Fe3 plus. This then acts as the electron accepting molecule and facilitates the oxidation of metal sulfate. So that is the basic mechanistic difference between direct and indirect oxidation. So when we talk about bioleaching or leaching in particular, you have heap or a dump leaching, you have in situ bioleaching, and you have bioreactor based leaching. The bioleaching can happen either in the site itself, or you have specialized reactors where you want this bioleaching to happen. So we'll compare all these three. So in case of setup, for heap or dump leaching, you will need an open heap. In situ bioleaching, you will have underground operations, and in bioreactor, you will have to build a controlled reactor. In case of bioreactor, as we all know, it's going to be highly in control. In situ, you can moderately control in situ, whereas in heap or dump leaching, you will have very limited control. There is high infrastructural requirements in bioreactor based leaching process, and for heap or dump leaching and bioreactor leaching, you will need excavation. Like you have to excavate the ore from that place and form a heap or take it to the bioreactor, whereas in situ there is no need for any excavation. So when you have to deal with low grade ores and tailings, you go for heap or dump leaching. When you are going to treat with ground ores or concentrates, you go for bioreactor based leaching. And in situ leaching, as we saw in one of the previous slides, is established for uranium extraction. And the processing time is going to be very high for in situ bioleaching and heap or dump leaching, whereas in bioreactor you will be able to control the kinetics of the process, so it will be in a controlled time frame. So the metals can volatilize, or they can escape from the system. So the risk of environmental exposure is going to be very high with heap or dump leaching and in situ leaching compared to bioreactor based leaching. And the operational flexibility is very low for heap or dump leaching and very high in case of bioreactor based leaching. In in situ, it's moderate. So next comes the role of acidothiobacillus peroxidans in biohydrometallurgy. So this is one of the key microorganisms in biohydrometallurgy. It is a chemolithotropic autotroph, and it has highly versatile metabolism. It can perform multiple oxidation reactions simultaneously, Therefore, it is highly valuable organism in biohydrometallurgical studies. So iron sulfide oxidation, for example, this azothiopacilus peroxidans is capable of oxidizing pyrite to produce Fe2 plus and sulfate as primary product. And this reaction is fundamental to many biological processes as it generates iron species and the necessary conditions for metal solubilization. And next you have thiosulfate metabolism, where this acetothiobacillus peroxidans produces thiosulfate as an intermediate product, and it can then be subjected to direct oxidation to sulfate. And this capability allows the organism to completely mineralize sulfur compounds, maximizing the acid production and maintaining the favorable conditions for metal extraction. And next there will be iron oxidation, where the organism will aid in the conversion of or oxidation of Fe2 plus to Fe3 plus. And this oxidation capacity is what is making it effective in maintaining the environment necessary for metal solubilization. And the energy and carbon metabolism that is required is that we saw that it's a chemolithotropic organism. Therefore, it utilizes iron and sulfur compounds as electron and energy sources. It derives electrons by oxidizing these iron and sulfur compounds while also fixing carbon dioxide under aerobic conditions to produce organic matter. So, this though we have already seen how there is going to be this carrying out of direct oxidation process and indirect oxidation process. I'll just have it in the slide for you to look whenever I upload the slides. So we have seen that in direct oxidation process what happens. So finally this organism should have an integrated oxidation cycle. So this acetothiobacillus peroxidans operates through integrated oxidation cycle which combines both the direct and indirect mechanism. In the initial phase there is going to be direct oxidation of FeS2 to produce sulfate and Fe2 plus ions, and in the amplification phase this Fe2 plus is oxidized to Fe3 plus, and in the maintenance phase there's going to be a continuous cycle of Fe2 plus Fe3 plus conversion which maintains the oxidative conditions. In case of product formation, you will see that the sulfate production solubilizes the metal deposits, enabling the formation of metal sulfates like copper sulfate, zinc sulfate and cadmium sulfate. The environmental requirements for this process include acetothiobacillus peroxidans. As the name suggests, you can tell that you need acidic conditions. It's acidophilic bacteria. Therefore, the optimal pH range is 1.5 to 3, and it can function under mesophilic conditions, and it requires oxygen. So, it will work in aerobic environments for oxidative metabolism. So, with this, we end up completing about this metal remediation using microorganisms. I'll just wait for a couple of minutes to see if there is any doubts in this metal remediation using microbes or biological metal transformation and remediation. If there is anything, please post in the chat box. So the next part of this week's lecture had discussions on other biological removal methods. So one of which is the enhanced biological phosphorus removal process. So when we talk about enhanced biological phosphorus removal process, it is a biological wastewater treatment process, and it is considered as the largest biotechnological industry in the world. The biological waste treatment processes are essential for protecting human health and environment, and they serve multiple purposes, including energy generation, production of value added chemicals such as bioplastics and biofuels. So, the key features of enhanced biological phosphorus removal process is that it has enhanced efficiency. So, in conventional wastewater treatment, we can only remove 30 to 40% of phosphorus biologically, whereas in case of biological processes, we can achieve over 90% phosphorus removal. So, by just directly using this process, you can bring down the you can bring down the phosphorus content in the treated water within the permissible limit of 0.5 to 1 milligram per liter. You will have high phosphorus recovery. It not only removes the phosphorus from the system we are trying to remediate, but also it tries to accumulate the phosphorus into some useful form. So you will have high phosphorus recovery in while using this EBPR systems. So considering that you are able to efficiently remove it from the treatment system, and you're able to have it in a very amenable form, it's going to give you high economic gains. So the economic viability will be high. The process has become increasingly viable as a source of phosphate as prices continue rising due to increasing demand and limited global reserves. So the recovery of phosphorus from waste streams could satisfy 50 to 20% of world demand for phosphate rocks. So that was about the enhanced biological phosphorus removal. Again, why do we need to focus on removing phosphorus from the system? It has its own implications, one of which is the eutrophication. So it is caused by increased input of phosphorus and nitrogen from the wastewater treatment plants and other sources. And this is going to create a huge ecological imbalance. So ideally, when you have a conventional wastewater treatment plant, it's going to treat this UOD and remove the organic pollutants. But when you talk about handling phosphate and nitrogen contaminants, you have to tweak your system or modify it to do that. If it is not done, then the discharge from your wastewater treatment plant is going to have high concentration of phosphorus and nitrogen. So this again will go to some water system. So that water system is going to act as the sink for this phosphorus and nitrogen, and it's particularly going to affect uh, discharge points like lakes and rivers, reservoirs, etc. So what is the disadvantage and what is eutrophication is that because of the presence of high amount of nitrogen and phosphorus, algae will be able to rapidly grow in the system. So you have oxygen from the atmosphere, you have nitrogen and phosphorus, and you have moisture because that's a water system. So conveniently, algae can grow and thrive in the system for that matter. So if it's going to grow beyond a certain limit, what happens is the entire water system is going to be covered by algae. That is what you called as algal bloom. Okay, so it's able to rapidly grow and cover the surface of the water body. So what happens? This algae needs oxygen for growth, so it has also depleted oxygen. And inside the water body, you can find all this phytoplankton, zooplanktons, plants, fishes, many aquatic organisms. So now there is no way for sunlight to enter, oxygen is depleted, nutrients are low, and these organisms cannot grow. So this is the formation of algal bloom, you call it eutrophication, and the implications is the ecological imbalance that we were talking about. And again, you will have toxic compounds forming there, your water quality is going to greatly deteriorate. So this is eutrophication and its effect, and this is going to be caused by the presence of high concentration of phosphorus and nitrogen. So that is why you have to remove both of this from the wastewater discharge. So one strategy that we saw was this enhanced biological phosphorus removal. It can happen through multiple mechanisms. So specifically, now we are going to talk about polyphosphate accumulating organisms. So these organisms, uh, they are the key players in this EBPR systems, because they can accumulate phosphorus in terms of polyphosphate bodies within their cells. Like we saw about bioaccumulation, right? In a similar manner, uh, the microorganisms will be able to store phosphorus by in the form of polyphosphates inside the microbial cell. So by this they achieve up to 12% phosphorus content on a dry weight basis. Okay, ideally in non polyphosphate accumulating organisms, you will find phosphorus to be 3% in weight in dry weight basis, whereas in PAOs you can see that up to 12% of dry weight basis is going to be phosphorus, and that much of phosphorus in terms of polyphosphate bodies it can accumulate inside itself. So the metabolic characteristics of these organisms include that they transport the soluble phosphate inside their cells. They convert it into polyphosphate bodies. So polyphosphate means it's a polymer. So it's a chain. And that chain is approximately going to contain 40 to 100 plus phosphate molecules. And uh, the link or the polymer link here is going to be the anhydride bonds, which is similar to ATP molecules, but it is much longer, and it has dual metabolic capability. One the aerobic phase and another is the anaerobic phase. So in aerobic phase, uh, you have the growth of the microorganism, phosphate uptake, and storage as polyphosphate body. So it's going to form the polymer using by catalyzing the anhydride bond formation, and then it's going to store it. In the anaerobic phase, on the other hand, the substrate uptake and formation of polyhydroxyalkanoates is going to happen, and the growth is going to be stunted. So ideally PAOs require oxygen. So when you have these organisms in aerobic phase, you will have the growth of the microorganism simultaneously with uh, polyphosphate bodies. Whereas in case of anaerobic phase, what happens? The growth is not that much, but you will have the formation of polyhydroxyalkanoates. So the energy metabolism here is under anaerobic conditions, they hydrolyze the polyphosphate to generate energy. So we all know that in ATP what happens? ATP conversion to ADP generates energy, right? So you are breaking a phosphate bond and you will generate energy. In anaerobic condition, the organism is in need of energy. So whatever polyphosphate body it has, it breaks it. So when the phosphate bond is broken, there is energy generation, and this further leads to the formation and deposition of PHAs, which is the polyhydroxyalkanoates. So in your treatment system, it is very important in which phase you have to have the microorganism. So in aerobic phase, it's going to act in a certain manner, and anaerobic phase, it's going to act in a certain manner. So based on your requirements, you have to maintain the conditions and have the presence of this microorganism. So when we talk about this enhanced biological phosphorus removal, we will have denitrifiers, which are facultative anaerobes. So they are mostly active under anoxic conditions and they facultative anaerobes. So which means they can still shift between being aerobic and anaerobic, and they thrive when the organic substrate concentration is higher in the aerobic tanks. So the metabolic strategies that these denitrifiers use is that they utilize the organic compounds to yield carbon and energy, and they reduce the nitrate to nitrogen gas under anoxic conditions. So this they do it by exploiting the redox potential under the anoxic conditions. So the contribution to phosphorus removal is that they remove nitrogen compounds and reduce the nutrient load. They create favorable conditions for the phosphate accumulating organisms by processing the organic substrates, and they also help in maintaining anoxic environment necessary for selective pressure. And the second player is the microtrix, which is the universal processor. So it is active in all tanks like aerobic, anaerobic, and anoxic, and hence it is highly versatile and it's called as a universal processor. It has a multiphasic function. So since it can be active in aerobic, anaerobic, and anoxic conditions, it has different functions, right? So in anaerobic condition, it can perform substrate uptake and utilization. In an oxic phase, it can perform nitrate to nitrite reduction, and the aerobic phase, it undergoes growth. It facilitates material storage and product hydrolysis. So altogether, it does exoenzyme production. So the exoenzyme production is very essential for hydrolyzing the polymers. And we saw that in the anaerobic phase, it takes up the substrate and utilizes it. Henceforth, it breaks down the complex organic matter into simpler form so that the other microorganisms can metabolically be benefited. And it is one of, since it is present in all the processes, it's going to build a system connection between the treatment processes. And third one is the streptococcus, which is the versatile fermenter. It has broad substrate range, so it can feed on different substrates. It is active under anaerobic condition and is capable of aerobic growth when needed. The primary functions include fermentation, metabolite production, and substrate supply. So fermentation includes conversion of complex organic matter into simpler metabolites. Metabolite production generates volatile fatty acids and other compounds, and substrate supply provides suitable substrates for other organisms. The strategic importance includes metabolites serve as substrates for Accumulibacter, which you will see in the next slide. It maximizes the substrate utilization under oxygen limited conditions, and it facilitates metabolic flexibility. So the last one is the Accumulibacter that we were talking about, which is the phosphorus remover. In aerobic phase, uh, it has profuse growth and phosphate accumulation. In anaerobic, we saw no, the growth is not very active, but it just focuses on substrate storage. So in aerobic phase, it is going to take up the orthophosphate from the wastewater, and it is going to utilize the PHAs that is present in the anaerobic phase, and will accumulate up to 12% phosphorus by dry weight. In anaerobic phase, it will release the orthophosphate from polyphosphate, take up the VFAs and acetate-like compounds, store substances like PHAs, and function as GOs, glycogen accumulating organism. So the last part of today's lecture is on nitrogen cycle. So we saw about phosphorus removal and all, and now we will see about this nitrogen cycle. So nitrogen is one of the most critical nutrients in the biosphere. We all know that we any organic compound is going to have carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus is again required. So nitrogen is very important. Biochemically, it is present in proteins, nucleic acids, and other constituents. And when we talk about plants, it is a very essential nutrients. We know NPK is very essential for plant growth. So we have first nitrogen fixation, where the atmospheric nitrogen is converted to ammonia, and this conversion is facilitated by nitrogen fixing bacteria, which makes the nitrogen biologically available. And next comes the nitrification process. It's a two-step oxidation process. So in nitrogen fixation, you have ammonia. Here this ammonia is oxidized to nitrite and then to nitrate. Nitration and nitration. So the two process together is known as nitrification. And next comes the assimilatory nitrate reduction, where the nitrate is reduced to ammonia and incorporated into cellular biomolecules as amino groups. And next, first you are assimilating it, and next is the dissimilatory nitrate reduction, where nitrate is reduced to ammonia and it is released outside the cell. It is not like incorporated into the cell itself. And in case of denitrification, you have the complete reduction of nitrate to nitrogen gas, uh, and the intermediates in this process include different nitrogen oxides. In NMOX process, which is the anaerobic ammonia oxidation, so NMOX stands for anaerobic ammonia oxidation. Here you have the uh, direct conversion of ammonia to nitrogen using nitrate as electron acceptor, and this usually happens in anaerobic conditions. So as the name suggests, it's going to happen under anaerobic conditions. So we'll just see about what happens in the nitrification process itself. So in nitrification, so here we saw, right, nitrification is two-step oxidation process. Ammonia is first oxidized to nitrate, which is the nitration step, and then nitrate is converted to nitrate, which is the nitration step. So first, when talking about nitritation, ammonia is going to get oxidized. And you have ammonia oxidizing bacteria that carries out this process. So the key enzymes that is used in the nitritation processes, ammonia monooxygenase and hydroxylamine oxidoreductase. So these two are the key enzymes that drive the conversion of uh, ammonia to nitrate. And the microorganisms that are capable of bringing about this conversion include Nitrosomonas, Nitrococcus, Nitrospina species. And next you have nitrite oxidation, which is the nitration process. So now you have converted ammonia to nitrite. This nitrite you will further convert it to nitrate. And here you have nitrite oxidizing bacteria, which includes Nitrobacter, Nitrococcus. These are bacteria, and you also have archaea like Nitrosocumulus, which can facilitate the nitrite oxidation to nitrate. The key enzyme that is used here is the nitrate oxidoreductase, and this process generates ATP through proton motive force. And finally, you have this denitrification process. So now what you have done, you have taken ammonia, oxidized it to nitrite and then to nitrate, and now you have to perform the denitrification process. It involves the stepwise reduction of nitrate to nitrogen gas through multiple intermediate compounds. So it's going to be exactly opposite. NO3 minus to NO2 minus, and then to NO, and then to N2, and then N2. So you're going to sequentially perform this reduction reaction. You're going to remove the oxygen in each of the steps. The enzymes that can bring about this are going to be nitrite reductase, and nitric oxide reductase, and dinitrogen oxide reductase. The electron donor here is the NADH2. So this is going to supply the reducing power because it is reduced, it will get oxidized itself and then help in reducing the substrate. So the heterotrophic organisms like Paracoccus denitrificans, some Pseudomonas species. So these are some heterotrophs that can perform the denitrification process. Thiobacillus denitrificans is an autotroph which can perform this denitrification process. So combining this nitrification and renitrification, the conventional system couples this process where ammonia containing wastewater first undergoes complete nitrification followed by denitrification. So that is how you completely remove this nitrogen from the system and send it to the atmosphere again. So ideally in wastewater you will have nitrogen in the form of ammonia. So first you will perform this nitrification. So ammonia to nitrate, and then this again will be uh, so whatever you have oxidized again you will reduce so that you finally end up having nitrogen gas which will escape from the water system itself. So this is how you perform nitrogen removal in wastewater systems. I hope this is clear. uh, so we almost have one extra live session and one more week to go about with the live sessions for this course before you take up your exams or complete the course. So can you please suggest like what could be the medium uh, in which we can do the other two sessions? Because initially we were following a pattern where we will solve some questions, we'll discuss some questions. So if you are interested in that, I can bring that back for the final two sessions that I'll be handling. Or if you want me to go about summarizing all of this again, I can do that also. Or if you need a hybrid model where I can discuss some topics and then we can solve some questions, if it has to go that way, that is also completely fine. Can you suggest if you would, what you would prefer? Okay, solving at least 30 MCQs. So I hope you have received the email, but yeah, we'll be having a session on next Tuesday, which will be the week 12 session, and then we will be having one more extra session. So in both these sessions, we will be having a 2-hour session, and I'll make sure that around for 1 hour 30 minutes to 1 hour 45 minutes we will only solve questions, and remaining some 10 to 20 minutes I'll take for any other clarifications or something. So I hope that is fine, and I look forward for you to join in that session, and that is how I can go about with having sessions with question and answers. Is that okay? Okay. So if you have any doubts, you can post it in the chat box. Otherwise, I think uh, we are done for the day. And thank you for joining in and patiently listening to the session. Thank you so much. Okay, then I'll close the meeting now. And before I close, I would like to reiterate that if you have any specific doubts in any of the previous lectures or in the course content or whatever I have taught, please come back in the last two sessions to clarify those. Thank you for joining in for today, and I'm closing the meeting now.